This sort of intervention that I'm going to be talking about does have some potential applicability to other neurodevelopmental disorders. There are some specific things about FASD that make this study particularly relevant to FASD, but some of the topics we're going to talk about are more broad than that. So I'd like to start out this presentation by talking about the brain a little bit, and I'm going to tell you a little bit about what we know about how alcohol exposure prenatally affects brain development and cognition by way of background for the intervention study that I'm going to talk about. So I'm assuming most of you know something about FASD. This is probably something you're familiar with, that prenatal alcohol exposure does cause neurodevelopmental disorders, and these are what some of the cognitive deficits look like in FASD. So children who are affected by prenatal alcohol exposure can have intellectual impairment, but they can also have a range of other cognitive difficulties. Things like sensory integration problems, difficulties with mood regulation and behavior regulation, problems with concentration and attention are very common in FASD, or what we call working memory. We see impairments in judgment and other executive functioning, so oftentimes individuals with FASD will have difficulty planning, organizing, sequencing their behavior. And we see impairments in adaptive functioning, so day-to-day kinds of self-care skills, communication skills in the real world. All of these things are affected by the brain abnormalities that we see in FASD. We now realize that this is a continuum, that these conditions have a range in severity, ranging all the way to full-blown FAS, but there's a spectrum, and there are many individuals who have these problems as a result of alcohol exposure who do not have the full diagnosed FAS, or fetal alcohol syndrome. Something like 70% of those individuals who have prenatal exposure have neurobehavioral effects or neurocognitive deficits, even though they may not have the diagnosis of FAS. So I thought I would just show you a couple of examples from one of our MRI studies. So we've imaged hundreds of kids with prenatal alcohol exposure over the years, and this is a particularly significant example. What you see on the screen on the left side is a typically developing child, 12-year-old male, and on the right side you have, for comparison, a child with FAS. And you can see probably immediately a few characteristics of what FAS does to the brain. The child on the right has, in fact, a smaller head and a smaller brain, so that's the most obvious difference. And this is something we see consistently. But this child on the right also has abnormalities in this big fiber bundle in the middle that we call the corpus callosum, where the arrow is pointing. So this is a major highway for information in the brain, communicating between the two halves, the right and the left. And this is abnormal in this child with FAS, especially in the back part of the corpus callosum. This child also has cerebellar abnormalities. So the cerebellum is this structure that looks a little bit like cauliflower. It's in the back of the brain, and you can see this child's cerebellum is very much underdeveloped. And all of this black space around the cerebellum is fluid that has filled in to fill the space where the cerebellum would normally be. So these are gross structural abnormalities. These are the kinds of things that we see in full-blown FAS. This is fairly rare, and we see this only occasionally when we look at the brain in FAS, in FASD rather. What I've been interested in for the past probably 8 to 9 years has been some of the more subtle abnormalities of fetal alcohol spectrum disorders. So we've focused some of our attention on the white matter in the brain. And the white matter is the connective wiring of the brain, essentially. The corpus callosum that I just showed you is a large part of, a large bundle of white matter, fibers. We know that alcohol interferes with this part of the brain, and it interferes by delaying the myelination of those fibers and by causing abnormalities in the myelination. Now, think about the wiring in the brain as being covered with an insulation that's made of fat, and that is why it is in fact white, because it's made of fat. The wiring is essentially wrapped with fat, and that allows the signals to communicate much, much, much more quickly. This interfering with this process, we believe, affects the speed of transmission of information in the brain. And it also occurs that alcohol interferes not only with the developing brain, but it interferes with the cells that will eventually take care of the white matter over the lifespan of the individual. So you have two sort of things going on. There's an initial hit during the pregnancy, during the development of the brain of the fetus, where the white matter is damaged, but then there's a lifelong impairment in the ability of the cells that take care of that white matter that will result in problems. This is a technical looking slide, and I don't want you to focus on all the details. I'm going to show you what to focus on. This comes from a study by Susan Smith at the University of Wisconsin, and this is particularly relevant to nutrition. So what she did in this study was to look at, this is an animal study, I believe this was mice. She's looking at the brain and how the brain wires itself and myelinates itself. So again, we're talking about this insulation over the wiring of the brain. And what she's showing in this study is that there is an interaction between alcohol and iron. Now, iron is something that we all consume in our food, and you may have taken iron supplements at some point in your lifetime, and we know that children can be affected by the mother's iron intake. So if a mother is iron deficient during pregnancy, this can cause problems for brain development. But what Dr. Smith showed in this study, if you just focus on the graph and look at the bars, the two vertical bars on the right side, you'll see an iron sufficient and an iron deficient animal. And you can see the difference between the iron sufficient and iron deficient, and if they've been given alcohol or not. What she essentially showed was that if the mother is iron deficient and is consuming alcohol, you have a much more significant effect on the white matter development. So this is an example showing us that nutrition can interact with the effects of alcohol during brain development, and that the results can be fairly bad if there's a nutritional deficiency during these critical windows, and then if there's a further hit from alcohol exposure. So we've been interested in studying children with FASD, and here's an example of one of our early studies where we looked in the corpus callosum of the brain. Again, this is a major fiber bundle that connects the right and the left hemispheres. And we found abnormalities in that part of the brain using a very sensitive MRI technique. And this is not just looking with the naked eye at the pictures. We're actually measuring some properties of the tissue itself. So we're measuring how well put together these white matter fibers are. And we found abnormalities in the back part of this corpus callosum, the same region where we sometimes see very, very severe abnormalities like the one I showed you earlier. So even in children who have a normal looking corpus callosum, where a radiologist may look at the pictures and say that's normal, even then we can detect that there are in fact abnormalities in those regions. So we think alcohol is affecting something about the white matter development, maybe the myelination. And we know that there's some potential there for interactions with nutrition. This is also a somewhat complex slide, but I really want to just show you this picture on the right. And this is a schematic, kind of a graphic depiction or a drawing of one of these white matter tracts that connects the two halves of the brain. So we have a right and a left hemisphere in the brain, and this blue sort of U-shaped set of fibers is showing you how the two halves are connected to each other. And in this study, what we did was we found those tracts using the technique that's really very good at looking at white matter, and then we measured the brain activity in the right and left hemisphere. And What we found is interesting in children who have FASD. We found abnormalities in the way that the right and the left hemisphere communicate with each other. And I will walk you through this. Again, this looks complicated, but I will explain it so that it makes sense. The figure on the left is, it shows you what's happening in the right and the left hemisphere of a child without FASD. So this graph on the left side of your screen is a control subject, or a child who does not have FASD. The blue line is the left brain, the red line is the right brain, and what you have along the bottom is time. So we're looking at 6 minutes of brain activity here. And the right and the left hemisphere should be communicating with each other very, very closely, and that is in fact what you see here. These lines, red and blue lines, are practically right on top of each other. The right and the left hemisphere are communicating exceptionally well in this child. And if you like math, you can see that the correlation here is about 0.89. So they're doing the same thing. Now look over to the graph on the right side of your screen, and you see that this is a child with FASD, and the right and the left hemisphere are essentially out of sync. They're not communicating well, and the signal is different in the two hemispheres over the course of that 6 minutes. And if you look at the correlation, you see it's much lower, about 0.5. So what this is telling us is that, and this is during rest, I should say. So the child is just laying in the scanner doing nothing. Even at rest, the brain is performing in a way that is not typical in a child who has FASD. And it comes down to communication difficulty between different parts of the brain. In this case, we're looking at the right and the left, but we could also just as easily be talking about a region in the back of the brain trying to communicate with the region in the front of the brain. The same process is occurring there. So I wanted to just give you a flavor for what we have been learning about the brain in FASD before I launch into this discussion of a treatment study that we've undertaken. So we know that there are structural and functional brain abnormalities in this disorder. We've known this for at least 40 years. There really are no questions that alcohol causes this kind of brain damage. It's very clearly established in humans, and it's even more clearly established in animal models where we can control everything. So we began to think about this issue a number of years ago, and whether there was anything that we could try to do to affect the development that is happening after we discover that a child has FASD. How do we think about an intervention for a neurodevelopmental disorder? This is different than how we think about treating a condition where we might just be able to correct a problem like diabetes by giving insulin. This is a more complicated kind of thing to think about because this individual's development is still unfolding. So this work is happening at the University of Minnesota. In fact, we've finished the first 2 phases of this study, and I'll tell you about both of them. At the time that we designed the study, we chose to focus on this because the only available treatments up to this point have been behavioral treatments and educational treatments, which are effective in FASD, but which are few and far between for the first problem, that there just aren't very many, and that they can only cover limited range of aspects of FASD. This is a condition that, as I mentioned at the beginning, affects lots of areas of cognitive functioning. So we wanted to try to do something that would be a broad-based intervention that would help brain development more broadly. And there's some preclinical work or animal research that has taken place over the last 10 to 15 years that suggests that we might be able to do something with nutrition that could help brain development. And we really decided to focus our intervention on one part of the brain called the hippocampus, and I'll tell you why. So the hippocampus, you can see in the picture here, is structured deep within the brain. It is kind of a horseshoe-shaped structure. It's on both sides of the brain. In fact, we have 2 hippocampi, a right and a left. And there have been a number of studies over the years in animals that have shown that alcohol exposure can damage this hippocampus, and maybe it damages it more so than other structures. It results in fewer neurons in the hippocampus, so that structure is a critical structure for things like memory, and what we see is that it actually is smaller and has fewer neurons, and the neurons that are there can be damaged. It also affects the neurochemistry of the hippocampus, so alcohol will cause changes in the way that the chemical communication takes place in the hippocampus. Now in humans, we have seen, in fact, that the hippocampi are smaller in children who have been prenatally exposed. We've seen that in our data, and a number of other research groups have published the same thing. We've also seen that the left and the right are imbalanced. The left and the right hippocampi are imbalanced in FASD. Normally there's an asymmetry where the left hippocampi is different size slightly than the right side, and that may have something to do with the language processes being more important on the left side. And so that asymmetry is disturbed. We see a different relationship between the left and the right in FASD. And we also see these differences when we do functional MRI studies. So when we're asking children to do tasks and we look at how the brain is activating itself, what parts of it are using energy, we can see differences in the child who has FASD. So we know the hippocampus is affected. Hippocampus is very sensitive during certain developmental windows, but there are windows really at multiple different time points in pregnancy. So this is not limited to one specific time point in the pregnancy. In fact, we know that the hippocampus keeps developing. So after the child is born, well into the 3rd year and even into the 4th year of life, the hippocampus is really undergoing a big growth spurt. The whole brain is really growing essentially at a rapid rate in the first couple of years, but the hippocampus is one of those structures that is still developing quite a bit even into the 4th year of life. So we thought there's a potential opportunity there. If we can find children who are still at that young age who we know have been exposed to alcohol, maybe that's a time point at which we can intervene. Here's some data from one of our studies just showing you that in fact the hippocampus volumes, the size of the hippocampus is smaller in children with FASD, and that's even when we correct for the head size. So we know that their brains are overall smaller, so we control for that when we do this analysis, and we still find that the hippocampi are small in kids who have FASD. Now there are a tremendous number of studies looking in mice and rats that have demonstrated not only that alcohol affects the hippocampus, but that there are things that can be done to change the way alcohol affects the hippocampus in the developing brain. A number of the studies that have formed the basis of this research have come from a laboratory at the university, or at San Diego State University, where Jennifer Thomas and her research group have been doing this work for more than 10 years. And what they have done is given various agents to these animals to see if they can change the way alcohol affects the brain development. And in this case, we're going to be talking about the studies they did with choline. And way back, as far back as 2004, Dr. Thomas and colleagues have shown that if you give choline, which I will describe in a minute, to animals, you can attenuate or reduce the negative effects of alcohol. So they started out doing this work by giving the choline to the mothers, and they would give alcohol to the mothers when the fetus was forming, and then they would give choline and to half the animals and compare, and what you see is that the choline reduces the damaging effects of the alcohol. That's interesting, but that's kind of hard to translate into a human intervention, because you would have to basically be finding mothers who were actively drinking in order to give them choline. So Dr. Thomas's group continued this work with the animals themselves, giving the choline to the animals themselves to see whether that could have any effect. And in fact, what they saw was that they could reverse some of the negative effects of alcohol in those animals who had been exposed. And they did this all the way out to day 30 in the rat, which is equivalent to a younger child. We're not talking about infants now, we're talking about probably children in the preschool age range. And when we translate what's happening in the hippocampus, we think that the kinds of changes that are seen in these animal studies could translate to the first 4 years of life in humans, maybe even into the 5th year. So the kinds of changes that they saw, the kinds of improvements, were in things like memory, or what we call delayed discrimination, behavioral changes in the way that these animals acted in certain situations, spatial learning, being able to learn things visually, spatially, and they discovered that there were some differences in effectiveness depending on when you give this. Earlier is better. So what is this nutrient that we're talking about? Choline is an essential nutrient, and we've really only known that since the late 90s. All cells require choline, and they will die by a process called apoptosis without it. So you have to have choline. If you as a human are mildly deficient in choline, you will have some temporary organ failure, and you will have difficulties with hypertension and memory. And it'll make sense why memory is involved here in just a minute. The cells need choline to form the cell membranes. So all cells use choline as a component in the structure of the cell. And the cells also need it for metabolism, or their basic energy processes. It's also a building block or a precursor to something called acetylcholine. So this choline that's in our diet and in our food that we're going to talk about is actually important to one of the major neurotransmitters in the brain. It gets turned into this chemical that acts as a neurotransmitter. And it's also an important methyl donor, which is a little bit technical, but basically this has to do with how your DNA gets translated into proteins. So how your genetics gets turned into actual cells, proteins. We have the capacity to make choline, so we all make some of it, but we need to eat it as well. And in the U.S., you can see by these numbers that we don't eat enough choline on average. Typically, an adult should be eating about 550 milligrams. The average adult in the U.S. eats about 312. You see the numbers for kids there under the age of 8. They need to be eating some as well. And interestingly, pregnant women need to actually consume more choline than the average adult. They probably need to be in the 700 plus milligrams per day range, because the fetus actually robs the mother of her choline. So not only are U.S. adults not eating enough choline, but U.S. pregnant women are probably not eating anywhere near enough choline during pregnancy. Choline is important for formation of the nervous system, and if you're deficient in it during a brain development stage, you will have high rates of neural tube disorders, and it's tied in with folate. So you may not know this, but you are all being supplemented with folate or folic acid in your food. If you eat anything that has enriched flour in it, you're already being supplemented with folate, because that's a public health intervention that reduces neural tube disorders, things like spina bifida. Okay, so here's more evidence that we know that choline is important for development. Pregnant women will become depleted, as I said, because the fetus takes it from the mother, and it's because it's so critical for brain development. The fetal levels are actually higher than the maternal levels, so the fetus has its own circulation system. And we know from looking at breast milk that this is important not only during the time when the fetus is inside, but also after birth when the fetus is being fed, hopefully with breast milk. And this is evidence that choline is important. It's very high in breast milk, especially at the beginning stage of breastfeeding. So what does it do? Well, one of the mechanisms for choline is that it affects the hippocampus directly. And I'm not going to explain this graph too much other than to say that there are some basic processes that happen in cells in the hippocampus that form our memories. And it has to do with how the cells fire electrically. And when you give an animal choline, you change the way that those cells in the hippocampus fire, and thereby you change the performance of that brain structure. So it is able to do memory in a way that it was not able to do without supplementation. So these studies have been done for many years now, and we know that both things are true. If you are deficient in choline, this hippocampal functioning will be abnormal, and if you supplement choline, you will improve the functioning of the hippocampus. And these are permanent changes. So this is the really interesting part, is that if you affect the hippocampus at an important stage of development during pregnancy or during gestation of the fetus, these are permanent structural changes. So how you build a brain in the process of when the child is a fetus, that brain will be affected throughout its lifespan. And there have been animal studies using these methods that show that if you supplement these animals during pregnancy and then look at them when they are senior citizens, they actually have better functioning memories and less decline in memory in their senior years. So again, what a mother eats during pregnancy actually has lifelong implications for that child. So we took all of this information into consideration when we designed this trial, and we saw that there was very strong preclinical evidence, very strong animal data to suggest that this could have an effect in humans, and very low risk in humans. Choline is something that we all eat, and so supplementing people with choline is not a high risk proposition. We felt like we didn't know the answer, so we could safely do this study because we weren't trying to do something that was already known, and we felt like there was a window of opportunity to do this because choline is actually making its way into more and more foods these days. It's now being added to baby formula, and it's being added to a number of so-called health food products, and we thought that this was an important time to do the study before choline becomes a very, very big part of our diets. Our goals were to focus on neurodevelopment. We wanted to really see if we could change the trajectory of the brain development. And we wanted to do this early because there's reason to think that this will work in 2 to 3-year-olds and 4-year-olds, but will probably not work in 12 and 13-year-olds. We wanted to do it in a randomized controlled trial because that's the ultimate design for really determining whether something like this works or not. You randomize people to receive choline or not, and then you test for differences. And we've done this in 2 phases. We've spent the first 2 years doing pilot work, or the beginning stages, and then we spent the last 3 years doing the second phase, where we really tested out the idea in larger numbers. When we first started this work, we didn't know some basic things about how people would react to this kind of a study. And we wanted to see whether, first of all, whether we could find populations like this, whether we could get enough children who are in the 2, 3, 4, and 5-year-old age range to do a study like this. We wanted to see how parents would accept a study like this, where a child would be randomized to either receive choline or to receive a placebo that is not thought to be effective at all. And that's the way that the study has to be done scientifically, but we didn't know whether parents would accept that. And we also didn't know some basic things about side effects and whether or not people could be compliant with giving choline to their children every day, which is what we did in this study. And we wanted to look at some basic safety issues as well. Even though this is a nutrient, anytime you give something to children, you want to be certain that you're monitoring the safety. So in fact, the first phase of the study was pretty successful. We were able to recruit all of the children that we needed to have good power in this study, statistical power, and we administered this to 20 kids, and the randomization was actually very well accepted by parents. So parents understood the risks that their children are facing in terms of cognitive development. They were very interested in not only seeing whether their child could benefit, but very interested in helping science advance, because they recognize there isn't much happening along these lines in FASD. And we had great families involved. 85% of the kids in the study completed the study, which is actually very high for a clinical trial like this. And we had no problems with people crossing over or supplementing their kids on their own once they learned about what the study was about. We had no differential dropouts, so it wasn't harder for the kids who were taking choline versus placebo, which is something we wanted to know. And we wanted to see whether the choline we were giving would actually increase their blood levels, and it did. It dramatically increased their blood levels of choline. And we saw very few adverse events in this study, and no serious adverse events. The second phase has been basically focused on increasing the size of the sample so that we can see what the effects are with larger numbers of kids. And this is really what you need to do to determine efficacy. And so we again did a double-blind randomized trial, so nobody knows who's in which group. I don't know which children are in the choline group and which are in the placebo group, and the parents don't know. There are statisticians who work behind the scenes to look at the data, and everything remains blinded until the end. And that's the way to do a study like this. We did this with children who were 2.5 to 5 years of age, and we followed them for 9 months. The 9 months was a bit of a guess as far as how long we would need to follow them to see changes in their development. We don't know that yet. We've sort of stuck with the 9-month approach because we think we can measure developmental change over that span, but we don't quite know that for sure yet. And what we expected to see was significant improvements in memory, processing speed, and cognitive functioning. So these are just some basics about who we included in the study. I'll just point out that we only included kids who had documented prenatal alcohol exposure. Usually that was by the parent report or through social service records. We included kids who had the full facial characteristics, but also those who did not, because we recognize that this is a spectrum disorder. And again, we included kids with growth deficiency and those without. That's a hallmark of FAS, but plenty of kids have cognitive deficits from alcohol who do not have growth deficiency. And so this was a range of children who have FASD, and we really designed it that way because we want to know if the treatment works differentially depending on how severely affected the child is. Those are some exclusion criteria there at the bottom. That's standard stuff that we do to rule out other medical conditions and things that could be causing problems with brain development. I won't spend too much time on this other than to say that there are some systematic ways of diagnosing FASD and of characterizing how affected an individual is. And so we do things like look at their faces, look at the structures that are affected by alcohol exposure, such as the lip, the upper lip, and the philtrum or the groove above the lip, the eye width, etc. We spent a fair amount of time developing a way to get choline into kids, and this was done in 2 arms. So half the group got choline, half got placebo, and we decided to give them 500 milligrams a day, which is a large amount of choline based on the fact that kids, we think, should be getting about 200 to 250. So we really wanted to maximize this to the point where it would be tolerable, but where everyone would be getting enough and then some. So it's a supplemental dose. We did this for 9 months, and because we're using this as a treatment, interestingly, we had to go through an FDA investigational new drug process. So this even though this is a nutrient, something that's in food like eggs, it is something that we had to treat like it was a drug in this case. And here are some of the outcome measures that we used. The first one is called the Mullen, and this is a developmental scale. It's kind of like an IQ test for kids. We give this, it usually takes about an hour, sometimes a little bit longer, to evaluate the child's language, visual skills, basic counting skills for kids who are a little bit older, memory, perception. It really measures a range of skills, and that allows us to get an idea about their global development. The main measure of interest is this, something called Elicited Imitation. This is a memory measure, and as you might imagine, it's not that easy to measure memory in 2-year-olds and 3-year-olds and 4-year-olds. But there are a number of procedures that have been developed over time. This one by a psychologist named Pat Baver, and it's a structured play situation. So we present the child with these toys that they've never seen before, and then we perform a bunch of sequences with them. So we do things with the toys in a sequence, and there's some examples here, like the playground example that you might demonstrate playing in a sandbox, riding a bike, walking a dog, etc. And there are other play situations that are kind of linked in time. So we have a camping situation where the child is shown a fishing pole, and then a fish is attached to the fishing pole magnetically, and then the fish is put on a fire, an artificial fire, to cook, and then the child in the camping scenario is put into a little tent and a little sleeping bag. And so the child has to repeat these actions, and kids are very, very good at imitation at this age, and they love to do imitation, so we get a pretty good measure of what they actually remember. And this is a very highly standardized way of measuring memory in kids. We're also measuring brain activity in these children. So we hook them up with an EEG cap, and we measure brain activity across the head, and we're looking for things like the signals of the memory processes. So how is the brain responding to these learning tasks that we're asking them to do? Trials like this are heavily monitored, so we do things like ask the parents what they're feeding the child with a diet recall survey. We measured the choline in their blood. We had these kids see our study pediatrician regularly, talk to the families every month by phone, and we had this whole set of data monitoring behind the scenes. So because we were blinded, we had these people behind the scenes who were able to actually look at the data and monitor the safety and tell us whether we should continue with the study or not. And it was also monitored by the FDA. So I'll just point out a few highlights here. We looked at 96 children overall for eligibility, and about 31 of them did not participate for one reason or another. So we randomized 65 kids, and you can see that about 30 to 34 kids ended up in each of the two groups. And we followed them, and very few were lost to follow-up. By the time we got to the analysis stage, we had 26 who had completed in the choline group and 25 who had completed in the placebo group. So pretty good adherence to the study overall. This slide has lots of numbers, but I'll just point out to you that we did not see major differences in the groups, which is good. We didn't end up with groups that were imbalanced as far as gender or ethnicity or FAS severity or those sorts of things. They ended up being pretty equivalent groups, choline versus placebo, which is what you want. You can see that there were some FAS, somewhere around 16 to 17%. There were some partial FAS, somewhere around 40 to 50%, and the rest had alcohol-related neurodevelopmental disorder. If you know IQ scores, you can look at these scores here and see that they're about in the low 80s, 83 to 84 for their overall IQ. So these are children who have some level of cognitive impairment. Compliance was great. They received choline, or they received a partial dose on at least 88% of the days, and there was no group difference. So again, it wasn't more difficult for kids to be consuming choline than it was for them to be taking the placebo, which is good. What we saw when we looked at their dietary intake was that there was no difference. So the parents did not alter the child's diet once they learned what the study was about. That's good. Their serum levels, or their blood levels, went up very significantly, 102% increase in their serum choline. So by giving them oral choline in this drink that we created for them, their blood levels went way up, and presumably the level in brain also went up. And then these other things are metabolites, or things that also go up when you give choline. So essentially what we gave them worked for their increasing their choline in their blood. The one side effect that we saw was one that we expected, and that was fishy body odor. When you take choline, it will come out in the sweat and in the urine if you're taking more than your body can absorb. And so we did see that in the choline group only, and that was a significant side effect, and that a number of children had it, almost half of the ones in the choline group. Not necessarily especially problematic, and it's something that we can manage, but it is a noteworthy thing. The results were that we did not see any changes in their overall intellectual functioning, and in fact we didn't really expect that we would see changes in overall intellectual functioning. And that's what this graph is showing you, that over the 9 months, both groups increased a little bit in their basic scores on the intelligence test, or the Mullen, and there was no difference in the slope or the trajectory of that. Where we did see a difference was in the elicited imitation, or memory performance, and this is in fact where we wanted to see the change. And this is a little bit complicated, but let's just focus on the graph on the left side, and this shows us a couple of things. If we look at the blue and the gray lines on this left figure, you can see that for the older children, these were the 4- and 5-year-olds, they were pretty flat. They didn't really show an increase in their memory performance, regardless of whether they were on choline or placebo. But when we look at the younger children, we did see the slope of this orange dotted line is more steep than the slope of the solid orange line. So in the younger children, the 2- and 3-year-olds, we saw an effect of the choline such that choline increased the memory performance for those young kids who were receiving it compared to the placebo group. This was over 9 months, and we don't know exactly when that change took place. We only measured their performance at 3 different time points: baseline, 6 months, and 9 months. And then we generate these lines from those 3 data points. So we don't know exactly when it happened, but we did see the effect there. Interestingly, oftentimes we think of more as better when it comes to things like treatments or interventions like this. But in this case, what the data are telling us, perhaps, is that the dose we were using might have been high for the children in this age range. And in fact, the kids who were receiving a smaller dose for their weight what actually seemed to perform best. So, and that combined with the fact that many of them had the side effect of fishy body odor suggests that the next stage of the study may want to use a lower dose and use a dose that's adjusted by weight. So we had gone with a standard dose across, and this data may be telling us that we need to adjust that dose by weight, which is what we're planning for the next stage. This is just showing you that the fishy body odor was common, especially in the kids at the far right here. This bar on the far right shows you that the kids who were receiving the highest dose of choline per weight, so these would be the smallest kids who got the dose, 100% of them had the fishy body odor. So again, suggesting that the dose we're using is definitely fully supplementing them, and then some of it's getting dumped out in their sweat and their urine. So this is where we're going to take this study next. We're going to focus on changing the dose and making it a flexible dose so that it will be for based on an individual's weight as opposed to just a standard dose across all people. And we're going to be evaluating some of the side effects again and some of the other metabolites of choline. And we're going to add some dimensions to the cognitive measures. So we're interested in not just memory, but we're also interested in some of those other cognitive skills like planning skills, organizing skills, future thinking skills. And we're going to do some follow-up with the children who were involved in the first study. So we want to know whether those effects are permanent or whether they were only a function of being on the choline itself. This is something we do not know yet. One more little tidbit I'll just share with you, and that is that we did look at some genes in these children, and there's a suggestion in the data that there might be a gene, there might be a relationship between having a specific gene or not and how a person responded to choline. There is a gene called PEMT, which is, we're actually interested in the mother's PEMT status, but this gene is involved in whether or not the mother can ramp up her choline production during pregnancy. And if a mother did not have this gene, she may have been even lower in choline during her pregnancy because her body was not capable of ramping up that production. And what we're seeing here is perhaps that the kids who don't have this gene or have the abnormality in this gene might not be showing as much of a, or might be showing a larger response to choline. So they may not have, they may be more able to benefit from choline because of the fact that they might have had this earlier hit in their development. Interestingly, I never thought that the children involved in this study were going to be deficient. We were going to supplement them based on the assumption that we were just going to take them where they were, and we had no real notion that they were going to be nutritionally deficient. But as it turns out, when we asked about the diets, and we did this in a systematic way, 84% of the kids had an inadequate level of choline intake, and they also had inadequate intakes of these other important nutrients. And this is probably for a number of reasons that kids with FASD have behavioral issues. Many of them have ADHD, are not likely to sit through meals. Many of them have some sensory issues, and so they might be more particular about certain foods. And what parents tell us is that they tend to be picky eaters. And so maybe this is not surprising that these kids have some nutritional deficiencies. But seeing this data really suggests to us that this is an area where we could really think even more broadly about attempting to correct their nutritional deficiencies at a very young age when brain development is still happening. I won't spend too much time on this other than to say that we're also looking at overweight status in kids, and one interesting thing that we found is that when kids with FASD reach adolescence, there may be some tendency toward overweight or obesity, especially among females, and there's a number of reasons why that may be true, some related to eating patterns, some potentially related to metabolism. So that's another thing we're exploring. I want to leave some time for questions, so I'm going to just briefly summarize here. We've learned that the brain, and especially certain structures like the hippocampus, are very vulnerable to alcohol, and that we think that alcohol impacting on the hippocampus early in development could have global cognitive effects. Because that hippocampus is so important in memory, if you disrupt it early in development, you may just disrupt a lot of other aspects of learning. And we're thinking now about optimizing neurodevelopment in these kids. And we're starting to see that it's possible to do these trials with supplementation, and that there may be some benefits from choline. Some other groups are looking at things like omega-3 fatty acids and vitamin D. But in general, these translational trials, taking this research from the animal world and trying it in humans, seems to be a reasonable approach, something that is feasible and potentially has high payoff. So I will stop there, and I'm sure that we might have some questions coming in.
Yes, indeed. Well, Jeff, that's an excellent presentation, very interesting information. Thank you so much. And indeed we do. So let's get right to it and see if we can get through a few questions before the top of the hour. First, a participant is interested if you would talk a bit more about the potential for choline to actually reverse brain deficits from prenatal alcohol exposure. And they write in their question that is it more a situation where choline may offset effects of alcohol if it's administered especially to the pregnant woman, or even reverse brain effects that could actually be described as healing the brain?
Well, the way I think about it is that we're trying to optimize what's left of development. So we know that the alcohol has interfered with brain development at a certain stage, and what we're trying to do by giving choline, and what we would be doing by giving other nutrients, would be to optimize development at that point going forward. So I don't think of it as necessarily reversing damage, but I think in the end, if you can optimize or maximize brain development, that maybe you can overcome some of that damage that was done by sort of increasing the rate of development, increasing, making that part of the brain as strong as possible, essentially.
And continuing on with regard to choline, how would you characterize, how far along the path are you and the researchers toward hopefully getting to a point of being able to actually recommend administration of choline either during the pregnancy and for young children?
Yeah, it's a very good question, and unfortunately these things move more slowly than we like, and it takes a number of years before we can get to a point where we really are sure that we know what we're talking about, and it takes this kind of trial to do that. We've now spent 5 years on this. I think we have some preliminary results, but I do think they are preliminary. I think we probably need another 5 years at least to do another trial that we're planning, and but I think we've learned some things, and I think our perspective has changed. One, I think we now realize that ensuring that mothers have adequate nutrition, especially mothers who are at high risk for drinking, is an obvious thing to do. Now, that doesn't necessarily mean giving them choline. It means optimizing their nutrition, making sure that they are nutritionally sound on all fronts. And likewise for children, maybe what this data is telling us is that we need to focus on identifying these children early and then optimizing their nutrition, making sure that they're getting help to bring their nutrition up to par. And you can do that with diet. You can do that with behavioral techniques for kids who are difficult eaters. You can do that with educating the parents about what the nutritional requirements are and where their child is, without necessarily just doing supplementation. So much of what I'm talking about here, I think, will ultimately be doable with diet. But you have to overcome some hurdles with kids who are difficult eaters, who are picky, those sorts of things.
Let me ask you sort of a question as really a compilation of several questions that are coming in now as you're talking, and you probably get this all the time. We have many parents now who are inquiring about children that are a bit older, and I know you mentioned in your presentation, you're looking at up to perhaps 3-4 years old as brain development continues. But what about older children, even into adolescence? Any possibility that choline might be helpful, and that might be something that we looked at in the future, or other nutritional substances or supplements that you have looked at all?
Yes, we don't have any data, so I can't tell you whether we should expect that older kids are going to benefit. From a pure science perspective of understanding how the brain develops and what we're trying to do with these individual nutrients, I have to believe that this is really going to be effective in the youngest kids, and even 2, 3, 4, and 5-year-olds are somewhat old relative to when this really would have its maximum effect. Ideally, we'd really like to be supplementing fetuses. I think that I don't have reason to think that choline is going to be effective in teenagers. However, I will say that we think that these dietary patterns probably continue of insufficiency, and I think there are plenty of opportunities for nutritional interventions in general for kids who have FASD to really help them on a number of fronts. But certainly the brain is developing into adolescence and even into young adulthood, and probably nutrition is a part of that. It's just a part that we don't really know much about yet. And I think there's every reason to think that if you have a child with FASD and they have dietary insufficiencies, that correcting those at any age can only help with whatever brain development is left to happen.
That's very helpful. You know, I loved your slide on that showed sort of the low correlation in the FASD brain when looking at the right and left hemispheres, communication, if I have that right. And we have a couple of people who've sort of asked about that, and sort of even just graphically looking at it, it sort of maybe suggests with that low correlation, you know, issues with attention and memory. How does that? We have a few people who've asked, how is that manifested in terms of behavior? Is there any thought there or anything that you've seen when you're looking at a brain scan that shows that lack of correlation?
Yeah, I would focus less on the left and the right. And I'll just say that we looked at the left and the right communication because it's a fairly straightforward thing to do with this kind of brain imaging. But I believe that the communication breakdown is really there across all regions of the brain. In fact, I have some other data to suggest that the overall communication throughout the brain is less efficient in children who have FASD. So how that manifests is most often in problems with high-level cognitive functioning, like attention and like perceptual skills, to some extent motor skills when it involves mixing motor skills with visual skills. So some of these higher level cognitive functions that require multiple parts of the brain to coordinate, I think, are the ones that are most likely to be affected. Practically speaking, kids who have these problems probably are slower processors when it comes to processing information. They probably can handle less information flow. They're probably more likely to become distracted, etc. And these are problems that we know are very, very almost universal in kids who have FASD.
I want to alert everyone that we may go a few moments past the hour here, but I want to, if I could, two more quick questions. One that just came in that asks, are there foods that are best or richest in choline to try to build into a child's diet?
Yes, liver. No, it's actually true. Liver is very, very high in choline. Of course, we're not going to get our typical American children to eat liver. Eggs is probably the easiest source of choline, and there are, in fact, 2 eggs is basically enough to meet the level of choline that we were using in this study, and even 1 egg is coming close to meeting the daily requirements of choline. It's not to say that children should be eating an egg every day, but perhaps we should be a little bit less concerned about cholesterol and things like that, and a little bit more concerned about overall nutritional balance and meeting nutritional standards. Meats are somewhat high in choline, so chicken, pork, beef all have relatively reasonable levels of choline. Wheat germ has choline in it. There is the USDA has put out a database, and my colleague Steve Zeisel has participated in developing that database of foods. And so if you use the everyone's favorite research tool, Google, and you search USDA choline, CHOLINE, you will probably come up with that database, and it shows the different levels of choline in different foods.
Before we go, Jeff, I'd love to ask you one other quick question, maybe a little off the topics of your presentation, but one of the reasons why you're in Washington, D.C. is for the meetings of CFAS, the Collaborative Initiative on Fetal Alcohol Spectrum Disorders, obviously a very important initiative and the world's largest consortium of research universities and institutes on this issue that is so important to us. So I wonder if you, well, I guess I should mention NOFAS actually has a very small role in CFAS in trying to promote some of the findings. But I wonder if you maybe share, you know, a couple of words about the importance of that initiative.
Yes, absolutely, and you can see the full spelled out name of CFAST here on the screen as one of our collaborative groups. So this is funded by the National Institutes of Health, the NIAAA, which is the Alcohol Institute, and so we obviously thank them for their support in both the work that I described to you and also the other work that CFAST does. CFAST is a collection of researchers around the world, many in the United States, but some elsewhere, who are looking at really multiple angles on the problem. Everything from trying to get better at diagnosis to understanding what happens in the craniofacial development in kids who have alcohol exposure, to understanding the brain and how the brain abnormalities manifest in cognitive difficulties. So we're studying things like attention span and how to differentiate kids with straightforward ADHD from those with FASD. There are genetics folks involved in CFAST who are helping us to understand how the genes interact with alcohol exposure. We know that not all fetuses are vulnerable in the same way, not all mothers are vulnerable in the same way, that there are genes that are critical to the development of this disorder. So it's really this broad spectrum of studies, including animal studies and human studies, looking at many, many aspects, trying to work together to move the field forward. And I'm thankful to be part of CFAST and thankful for the support of the NIAAA for this really, really outstanding collaborative group.
And your work is so important and representative of, I think one of the priorities of CFAST, which is to translate those findings into practical interventions and products, for that matter, that would have a positive impact and improve outcomes for those who live with fetal alcohol spectrum disorders. So, Jeff, you're terrific for doing this. Can't thank you enough. NOFAS is grateful to you and is just so pleased that we have a chance to collaborate with you. We hope to bring you back again for more great information for our audience. I would thank all the participants who were on the line today and remind you again that you can view this archive from nofas.org by tomorrow or so. If you have some questions, please go ahead and send those in and we'll see if we can forward those to Jeff, and if he has a moment, maybe give you a quick, either he or NOFAS give you a quick response. So, Jeff, thanks again, and everyone to join us next month in April for the next edition of the NOFAS webinar series. Thank you. Bye.
Bye-bye.